Welcome back. Today I'm going to review some of the key points in Chapter 2. As always, I urge you to view all of the PowerPoints, read the associated chapter in the text, and complete your discussion boards, quizzes, and other assignments on time. One of the key distinctions in the human nervous system is that between the central and peripheral nervous systems. The central nervous system is composed of the brain and the spinal cord. This is what mediates the information that comes in from and goes out to the peripheral nervous system. That means, of course, that the peripheral nervous system is everything in the nervous system that is not part of the brain or the spinal cord. This involves sensory inputs and motor outputs. As you can see in this chart, the peripheral nervous system is broken down into a number of parts that we'll go over in time. First, let's talk about the cells that make up your nervous system. There are two basic types of cells in the nervous system: neurons, which communicate information, and glial cells. Neurons communicate with each other through what are called action potentials. These action potentials involve transmission of electrical impulses down the axon of one neuron, resulting in the release of neurotransmitters into the synapse, where they then connect to receptor systems in the dendrites of an adjacent neuron. The axons are covered with a semi-permeable cell membrane. This membrane allows some substances to move easily through it. Other substances require energy to move through it, and some substances cannot pass through the membrane at all. Action potentials involve the movement of ions, which are charged particles, from one side of the neuron to the other. In between action potentials, the neuron is in a resting state where it is slightly more negatively charged inside the axon than out. During the action potential, positive ions move into the inside of the axon at a rate that changes the electrical charge so that the inside of the neuron becomes more positive than the outside. While both sodium and potassium ions are positively charged, during an action potential, far more sodium ions enter the axon than potassium ions leave. Some neurons have their axons covered with a fatty substance known as myelin. We call these myelinated neurons. Others are not so covered. The ones that are myelinated conduct information much faster because the ions enter and leave only through the nodes of Ranvier, that is, the gaps in the myelin coverage. In unmyelinated axons, ions must enter and leave at each point along the axon, which takes considerably more time. When an action potential occurs, this causes the release of neurotransmitters, which are small molecules that communicate between the presynaptic neuron and the postsynaptic neuron. On the surface of the dendrites of the postsynaptic neuron are a number of specially shaped receptors that receive the neurotransmitters. Each neurotransmitter has a specific shape of receptor that it can lock onto, and it cannot activate any other receptor. Some neurotransmitters increase the probability of an action potential in the postsynaptic neuron. We call those excitatory neurotransmitters. Others reduce the likelihood of an action potential in the postsynaptic neuron. Those are called inhibitory neurotransmitters. Over 50 different neurotransmitters have so far been identified for the human brain. A few of them that have major effects are listed here in the chart. Let's move on now to the structure of the brain. As seen in the image here, the brain has three major divisions. Immediately above the spinal cord is the hindbrain, which controls survival functions like breathing, heart rate, swallowing, etc. Above this is the midbrain, which has important roles in modulating movement. On the very top is the forebrain, which is involved in memory, speech, emotion, complex thought, and language interpretation. The hindbrain has three major parts. The medulla, which controls functions such as heart rate and breathing. Above it, the pons, which regulates sleep and coordinates the movements of the left and right sides of the body. And in the back, the cerebellum, which is essential for coordinated movements that we make all the time without even being aware of them. The midbrain is involved in modulating movement, in the reflexive movements we make with our eyes and the rest of our body. And it has an important structure called the substantia nigra, which is needed to initiate voluntary motor activity. When most of the neurons in the substantia nigra have died off, the result is a condition called Parkinson's disease, which is characterized by stiff movements and difficulty in initiating movement. The forebrain is composed of the cerebral cortex, which is on top, and the subcortical structures. The cortex is divided into a left and a right half. These two halves of the brain communicate through a structure called the corpus callosum. Each hemisphere is comprised of 4 lobes or sections. Let's look at each of these in turn and see what their main functions are. The prefrontal cortex is involved in those processes that make us uniquely human. Understanding social rules, attention, concentration, decision making, and the planning of language. At the very back of the frontal cortex is the primary motor cortex. This is where motor movements are initiated. Combining the prefrontal cortex with the motor cortex gives us the frontal lobe, which is colored orange in this photograph. Behind the frontal lobe is the parietal lobe, which is involved in touch sensations, particularly in the primary somatosensory cortex, and localizing objects in space. Underneath the temples are the temporal cortex, which is involved in hearing, memory, and understanding language. In the very back of the brain is the occipital lobe, which is where visual interpretation begins. The prefrontal cortex makes up approximately 30% of the volume of the human brain and is much more developed than in any other animal. Some of our first understanding of the functions of the prefrontal cortex came about as a result of an accident in 1848 to a young railroad foreman named Phineas Gage. Gage, shown here on the left, was injured when an iron tamping rod, which he's holding in his left hand here, was driven through his skull as a result of an accidental explosion. Amazingly, Gage not only survived the accident, but remained conscious and walked under his own power to a wagon in which he was driven to the nearest town. Though he recovered from his wounds, Gage was fundamentally changed by the accident. Missing parts of his prefrontal cortex, he had great difficulty in sustaining attention and remembering what it was he was supposed to do. His social skills were also somewhat impaired. Sadly, Gage died some years later as a result of seizures brought on by this accident. Let's now move on to the peripheral nervous system. As noted before, the peripheral nervous system consists of all of the nervous system that is outside of the brain and spinal cord. And the peripheral nervous system is broken into two main components: the somatic nervous system and the autonomic nervous system. The somatic nervous system contains sensory inputs from the periphery, that is, sight, sound, taste, etc., as well as motor connections from the spinal cord or the brain out to the periphery to initiate movements in the body. You can think about the somatic nervous system as being responsible for voluntary movement and conscious perception, those things that we're aware of doing. The autonomic nervous system, on the other hand, is involved in the involuntary, automatic functions of the body. It carries information from the internal organs and glands in the body and produces automatic responses to the environment depending on our needs at the time. It has two complementary divisions: the sympathetic nervous system and the parasympathetic nervous system. The sympathetic system, sometimes known as the fight or flight system, prepares the body for action, increases respiration and heart rate, dilates the pupil, and decreases digestion. The rest and digest system, the parasympathetic division, slows everything down. These two systems work in concert to maintain the appropriate level of activation in the body for the organism's current needs. Along with the neurotransmitters, another group of chemicals that are essential for the functioning of the human nervous system are hormones, which are chemicals secreted by one gland that travel through the bloodstream to a target somewhere else in the body. Each of your cells contains thousands of genes, which are segments of the DNA molecule that code for the production of proteins in the body. You inherit these genes from your parents. All genes are not active all the time, nor are they active in every cell. Epigenetics is the study of how the environment activates or turns on genes. Genes do not produce a protein unless activated by some external stimulus. And by external, I mean something outside of the gene. It can be within the cell, outside of the body, or even outside in the world through its interpretation by the human brain. One of the really amazing features of the human brain is its ability to adapt, change, and take over functions for damaged parts. When we have damage to the brain, even as extensive as that's shown in the photo, other areas of the brain are sometimes able to take over the functions of the damaged part. This process is called plasticity and is particularly effective in younger people. As one grows older, the plasticity of the brain tends to decrease. I hope you have enjoyed our brief tour through the human nervous system.